Good morning or good afternoon from wherever you are. Welcome to the Harmful Algal Bloom webinar series. My name is Laura Freskin and I'm from the Iowa Water Center located at Iowa State University, and I'll be your moderator today. The webinar is hosted by the Algal Bloom Action Team, of which Iowa State is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resource Research Institutes, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets and frequently asked questions on HABs topics. I encourage each of you to visit our website, which I am putting in the chat directly after this, and there you can explore some of the resources we have to date, including a what you should know fact sheet, a HABs frequently asked questions database, and recordings from previous webinars, and our virtual HABs Research Symposium, which was held in January. So before we get started, just a few housekeeping items. We will have a Q&A session after our speaker presents today. So please list your questions for our speaker in the Q&A panel, and we will do our best to address all of them following the presentation. If there is a question already posted to the Q&A panel and you have the same question, you can upvote the question and we'll be sure to get those answers to the most popular questions during the session. Today's webinar is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you are having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat and we'll be happy to assist you as best we can. Today's presentation is titled Toxins Associated with Freshwater Harmful Algal Blooms and Their Mammalian Health Effects. So this presentation is by Donna Hill from the US EPA. Donna has practiced small animal medicine for 20 years and has been with the US EPA for nearly 15 years. The past 12 years, she has focused on rodent toxicology studies and studying freshwater cyanotoxin health effects to contribute to the EPA's database for establishing health guidance. Donna works in the Office of Research and Development in Research Triangle Park, North Carolina. So we are going to start our presentation. I will stop sharing my screen, and Donna, you are welcome to start sharing yours.
Thanks, Laura. Let me see if I can get everything selected properly here. Can you see the presentation?
No, I cannot, not yet.
All right, let's try this. Can you see it?
So the share screen on, there we go. It looks like it's loading. Perfect.
Okay, great. Thank you so much. Yeah, I really enjoy the HABs webinar series that this group hosts, and I'm glad to have the opportunity to be here today. And I do mouse toxicology studies on cyanotoxins, and my goal is to try to produce data that can be used by other people in the EPA towards development of human health guidelines. This is my disclaimer. This presentation does not necessarily represent the viewpoints of EPA. And I think most everybody on the call knows what a HAB is. It's a large rapid growth of cyanobacteria or algae, and it can be in fresh, marine, or brackish water. I'm focusing on the freshwater HABs today. And they are a natural phenomenon, but seem to be increasing. It's a debate whether it's increased incidence or awareness. I think it's probably a little of both, but in the distribution, frequency, duration, and severity. So in freshwater, the contributor for toxins are the cyanobacteria, and a lot of the species are non-toxic, but it seems like a lot of the negative environmental impacts seem to favor the toxic varieties. And historically, we've been most familiar with the planktonic blooms, the ones you can see the scum, you can see colors, but I think most everybody's probably aware too that the benthic blooms, the ones that grow on the bottom attached to sediment, rocks, and plants, they are getting a lot more attention these days. And why do they occur? Increased nutrients, warm temperatures, low water flow, optimal pH. The nutrient pollution contributes excess of nitrogen and phosphorus from multiple sources. And climate warming. There are several characteristics of climate warming that seem to encourage cyanobacterial growth. The increasing temperatures, storms causing more runoff, and then followed by drought. Warmer temperatures and low water flow, increased carbon dioxide. This is a view of the Spokane River in 21. They had drought conditions, and there were a lot of problems with HABs that year. And this is a typical sign we expect as a warning for a harmful algal bloom. It, usually has a picture of something that looks like green paint on the water, or it says it can be different colors. But that is not the only type of thing we need to worry about, because this also is a harmful algal bloom. In Zion National Park in the summer of 2020, they had low rainfall. The Virgin River had low and slow flow. And you can see where the boy is standing, the water looks great, it's nice and clear, but the dog has noticed something over to the side, and those are benthic mats that have bubbled up to the surface as the oxygen is formed, and it tears the mats loose, and they tend to gather along the banks. So this was a microcoleus bloom producing a lot of anatoxins, a lot of dihydroanatoxin as well as anatoxin. And the dog orienting to this was not alive an hour after this photograph was taken, unfortunately. So the toxic effects, they affect fish, birds, and mammals. And in mammals, we include marine mammals, because of freshwater toxins can come down to the land-sea interface and have effects. Wildlife, pets, livestock, and humans. They're suspected in unusual mortality events of marine life. And a lot of this are the marine toxins, but some of it too are the freshwater toxins that make their way down to the marine environment. And these are probably the key cyanotoxins that most people monitor for. We've got anatoxin and saxitoxins, or neurotoxins. Microcystins are often we assume nodularins and microcystins act similarly because of their structure. The nodularins are mainly in brackish water, and microcystins in freshwater, and they have their primary target is the liver, and cylindrospermoxin targets the liver and the kidney. Now, in harmful algal blooms, we know this is just a very, very small part of the bioactive compounds that are available there. And, some of them have been identified, and probably a lot of them have not. And very few of them had toxicology work done on them. One that you may have heard of, the lipopolysaccharides, are also referred to as endotoxins. That is something from the cell wall of cyanobacteria, and also gram-negative bacteria. And that causes an inflammatory reaction. And, you got to wonder if in a bloom where there's so many different things, I mean, you do have bacteria and cyanobacteria and, lots of other organisms, what are the health effects really coming from? So this is off the EPA website. These are common health effects seen in humans, and I guess with the blooms that are predominantly one of these toxins. And microcystin and cylindrospermopsin, I mean, they're kind of generalized, non-specific symptoms that could be related to a lot of different types of illnesses. You've got GI signs, respiratory, just what they call generalized malaise, headache, maybe muscle aches, being tired. And then for the antitoxin, since that is a neurotoxin, you've got more of the neurologic signs, tingling, numbness, you can have tremors. In animals, death is caused by respiratory paralysis of the diaphragm because it affects a specific type of neuroreceptor, and it's at the neuromuscular junction is what affects the muscles and the diaphragm, but it can also act on the central nervous system. So we have a national reporting system that CDC has. It's OHABs, it's One Health harmful algal bloom system, and the One Health approach, which I feel like most people are probably aware of. It's using multiple disciplines to approach health concerns that can affect humans, animals, and the environment. And so this is set up, it's a reporting system, it's voluntary, and it's open for public health departments and state agencies associated with them in reporting the HABs events. The most recently published report is from 2020 on this OHABs. And so in 2020, 13 states reported 227 harmful algal blooms. And I believe 72% of those were just routine monitoring that caught them. And 45%, the water was murky and discolored. 20% the water was clear, but I guess the good news is that 97% had some visible scum or mats. So I mean, that is at least consistent between benthic and planktonic. So that's kind of a relief to me that there's some indication that people can look for. There were 95 cases of human illness, 1,170 of domestic and wildlife illness. 51% had toxins, and microcystin was the most common, followed by antitoxin. And as you go down the list, like saxitoxin and cylindrospermopsin seem to like flip back and forth on being the third and fourth most common. GI and the generalized symptoms were the most reported illness with humans and animals, and 42% of humans also reported effects to their skin. For the human reports, 23% occurred in national parks, 22% occurred in one event, and there was one death, but it was with paralytic shellfish poisoning saxitoxin. I believe that was in Alaska. The domestic animals are usually represented by pets and livestock, but in 2020 it was just dogs. And in wildlife, there's always some marine mammal representation. But this year, there was one large event where 1,000 carp were killed in one HAB. So exposure can occur through drinking water, recreational water, dietary supplements, and food. And with food, it's not just the creatures that live in the water that may be contaminated, but also if contaminated water is used for irrigation of crops, it has been shown that the cyanotoxin can be taken up by the vegetable. And this is just a little comparison of routes of exposure depending on the situation. So whether you work around the water that's contaminated, you're drinking it or recreating in it, you can get exposed, multiple ways. I put this medical intravenous, it's kind of like it always has to be mentioned because it did happen and it was horrible. In Brazil in 1996, 131 patients were exposed to cyanotoxins in their dialysis solution. And 116 became ill, and 100 had acute liver failure, and 52 ended up dying. And they identified cylindrospermopsin and 3 different microcystin congeners. It was LR, YR, and AR, if I remember correctly. So the EPA does not have, there are no regulations for cyanobacteria or cyanotoxins in drinking water. But we do have health advisories for microcystin LR and cylindrospermopsin in both drinking and recreational water. The World Health Organization also has guidance values. They have it for microcystin, anatoxin, cylindrospermopsin, and saxitoxin. These values here are EPA's guidelines for recreational water, and they're used in 2 different ways. One is if you exceed these on any one day, it's calls for a swimming advisory. But if you repeatedly exceed these values for more than 3 times in a season and more than 1 year, have it reoccur, it does, you can use it as a water quality standard, and it would show impairment of your recreational water. The drinking water health advisory is the maximum amount of exposure for a 10-day period, and EPA felt it was important to distinguish between adults and children 5 and younger because they have a higher water intake based on their body weight. So, you know, just by that virtue, it can make them more sensitive. The most recent WHO guidelines came out in 2020, and they have recreational and drinking water for the 4 toxins. Some are acute, some of them are lifetime. But they have that for saxitoxin, anatoxin, cylindrospermopsin, and microcystin LR. And the WHO lifetime exposure of microcystin LR is 1 microgram per liter. And for anatoxin A in drinking water, it's 30 micrograms per liter for an acute exposure and double that for recreational. It is difficult a lot of times to find the cause, you know, and confirm it. You can have a high suspicion, but with the symptoms of illness that humans and animals may experience after being exposed to a harmful algal bloom, like we already mentioned, they're nonspecific, they're very general, and it may take a while to connect the dots and think about, oh yeah, it could be, you know, the water I was exposed to. And with any delay, you have problems identifying it both in the patient, because the toxin, evidence of the toxin may have already left the bloodstream, not be in the urine, and in the environment especially. It's you rarely have a sample at the time of exposure, and it is such a dynamic environment. It is hard to know if you're getting the exact representative sample of what the animal or the human was exposed to. So we end up relying on animal toxicology a lot because, you know, human data would be the gold standard. The problem with that is that, you know, humans have so many other confounding factors, like their health status, their age, you know, their diet, so many things. It'd be hard to kind of tease out exactly what was caused by the cyanotoxin. Animal studies are limited by the cost and availability of the toxin. And that's why in this lab, we use the mouse. We use the smallest mammal we can to, you know, be conservative with the amount of toxin required for these studies. It takes much, much less toxin to do an injection called intraperitoneal, and that's just an injection through the skin and the body wall into the free space in the abdomen. And because that's lined with blood vessels, it gets taken up into the bloodstream very quickly, and it takes much less. And those can help with or the in vitro can help with mechanism studies, try to figure out certain mechanisms of toxicity with the particular cyanotoxin. Now, in general, our toxicology studies, for it to be used as a human health guideline, it has to use the same route of exposure that they're doing the guideline for. So we do oral in this lab, but there's also, inhalation and dermal as well. Lots of different things we do, and neurotoxins would be different from liver toxin of how we would set things up. But in general, we observe the animals after we treat them. There's different function tests like neurobehavioral tests, respiratory function, cardiac, just different things we can do to evaluate them. And then when the treatment is done, necropsy or the post-mortem exam is very, very important and gives us a lot of information. We collect tissues, we collect blood, and the blood, we can do serum chemistry analysis, just like similar to what you get when you go to your doctor for your general health exam. It's just liver and kidney function, your glucose, we do a lot of the same things with the mice. There's also other biomarkers that can be checked that can't be included in this serum chemistry analysis, but they have separate tests that we can look for different biomarkers for different problems. We can take tissue and do histopathology or specialized immunohistochemistry staining to look, that helps with mechanisms sometimes as well. Gene expression, metabolomics and transcriptomics. Those last two, they create a lot of information, and we're still kind of early in the process of learning how to make use of it all. But the effort's always there to try to get as much information as possible. And to me, the mouse is a hero because of this. I just want to go over some of the work that we've done in this lab in the past several years. To give you an example, we did a 90-day cylinderspiroxin study. The mice were dosed orally for 90 days, and some relatively small doses. 75 was the smallest dose, and 300 was the highest dose, and that's micrograms per kilogram once a day. And at the end of the 90 days, the mice appeared normal. They looked fine. They were eating, they were gaining weight, but when we did the test, we found out that none of the doses of cylinderspiroxin were in no effect. We did not find a no effect level. Everything affected, we had liver and kidney effects. So this is just one of the graphs I used as an example. This is the histopathology scores for the liver. And we have female and male. And then, these three bars are, for the dose groups. And they all had adverse effects. So California's EPA has used this study as an interim notification level for drinking water for cylinderspiroxin for up to 3 months exposure due to liver damage. We've also spent quite a bit of time looking at microcystins, and this we've used a lot. It's such a useful study, but this is looking at different microcystin varieties. I call them congeners in the Midwest states. And you see a lot of representation of red and yellow. The red is microcystin LR, the yellow is microcystin RR. We also see green, which is microcystin LA, and this blue is YR. There's also some LY and LW and LF were found in this survey. And I think the biggest surprise is one lake that didn't have any. Anyway, we used this information to help guide our microcystin study. What has been found at the time in the US and is most common. So when we started our evaluation of microsystems, very few had been tested in animals. Only 2 had been tested by the oral route. And I think that was LR and RR. And so we ended up comparing 10 of the microcystin congeners. We started by doing kind of a dose response curve on microcystin LR, and we wanted to find a dose that it was definite toxicity, but had room on the scale of toxicity for other congeners to show less toxicity or more toxicity. And so we ended up choosing 7 milligrams per kilogram as our dose. And then we took all the 10 congeners, which included the LR, and we dosed at 7 milligrams per kilogram, just a single oral dose. That's all we did. And then evaluated the mice with necropsy 24 hours later. The congeners we looked at were LA, LY, LF, LW, YR, WR, RR, and 2 demethylated RRs that were sent from Germany. They saw that a lot and they requested those. And so a lot of the things I listed in my toxicology endpoints, I mean, we did with these mice, it was relatively basic. And we found that in relative toxicity, the LA was more toxic than the LR, followed by LY and YR, and RR was virtually, hardly caused any change. So we were happy with that information, but it also showed that, prior to this, a lot of the toxicity was based on in vitro tests and whether it was more hydrophilic or hydrophobic. And it was thought that, the hydrophobicity gave the order of toxicity, being more hydrophobic, being more toxic. But when you give a microcystin orally, these are huge molecules. They have to be transported wherever they go. I mean, you take them in, but to cross any wall, they have to be transported. And so depending on what amino acids are connected to them and how bulky they are can affect how they are transported and how they affect their target. And this is a comparison of something called ALT. It's a liver enzyme that is leaked into the serum when liver cells are injured. And we have all the different congeners lined up. The solid lines are the males and the hatched lines are the females. And in this particular comparison, the LA, LR, and LY were significantly higher than control. And those were the only 3 in this comparison. What was really interesting is what MCLA did to the bilirubin. The bilirubin is made in the liver. It's, if you have too much of it, is what causes jaundice, that yellow in your eyes, and mucous membranes can have a yellowish tinge. It's often seen with end-stage liver disease, but this was just a single dose. Some of these mice weren't even looking, well, at 7 they were, but at lower doses, later on when we did studies, they didn't even seem sick and they could be jaundiced. So there's definitely something happening with LA and in the liver that's different from the others that we tested. And of course, you can tell from this, the females seem a lot more sensitive. So we took the 4 most toxic congeners from that comparison study, plus the RR, because it's just so common, it needed to be evaluated, and tried to determine NOELs and LOELs. So that's doing a dose response, doing multiple dose levels of each toxin, and then try to find the highest dose that's not toxic and the lowest dose that is toxic. So that's the NOEL and LOEL. And the order of toxicity was still the same. It was LA, LR, LY, and YR. And again, we continue to see the bilirubin differences. Now, the way that the Office of Water has set up the human health guidelines, there's a lot of safety factors involved. And so the LA is still within those guidelines. So the drinking water and probably recreational water guidelines are still covering for LA. Where it may be a problem is when you're doing ELISA testing and you're doing LR equivalents. You know, you can overestimate if it's an RR bloom, but you can underestimate if it's predominantly LA, because that's going to be a little more toxic than LR. And again, it's the liver enzyme we're looking at. I just pulled out the male graph for this, but this is looking at increasing in this activity of this liver enzyme as dose increases. And you look to see where, at what level, dose level, the increases start. So the red line is MCLA, and it increases at a lower dose, and then that's followed by MCLR. LY and RR tend to increase about the same time here. And we took RR all the way out to 22 milligrams per kilogram, and it didn't cause any increased liver enzymes. It did cause some glucose changes, but it was not affecting the liver. So we ended up with, you know, these no L's and low L's, the no observable adverse effect level and the lowest observable adverse effect level. And those are published, so if you want to refer to those. But you know, we said that the low L for RR was at 22 just because we did see some glucose changes, but it wasn't liver. And for future studies, things that we have approved for our next research round, which we are currently in, it started October 1st of last year. The one I've been preparing for, it seems like forever, but I'm trying to make the most of it, is an antitoxin 5-day oral study in mice. And this is tough because it seems like the only endpoint for antitoxin toxicity is, you know, death, and we don't want that. We want to see what can happen from repeat exposure to antitoxin that's, you know, of course, non-lethal, and see if there are some other endpoints that we're just not aware of. So we're looking at male reproductive effects, of course, neurobehavioral, and we're looking for auditory effects. It can certainly affect some of the neuroreceptors involved with hearing, but we've got a lot of different things, a lot of biomarkers, and a lot of things we're collecting in this study, and I hope to start that this spring. So stay tuned for that. There's not a lot of toxicity on nodularin, so I was going to do just an acute oral nodularin R toxicity study in mice, and then if I have all that done, I'll do a 90-day microcystin LR study in mice. I think the low dose, long-term exposure information can be really important because that's primarily probably what people are most exposed to and not even realizing it. This group of people over the years have contributed to all this work, and they are now all gone except for me. So we need more people working on this. It'd be nice. And that went a little bit faster than I thought it would, but that's what I have. So if you have any questions, I'll be glad to take them.
Oh, that's great, Donna. Thank you so much. We've got a lot of questions that have come in, definitely. So if you want to stop sharing your screen. All right. get things transferred over here. I want to put this up because in case we can't get to everybody's questions before the end of the time, you've got Donna's email here, and you can specifically shoot any questions to her that you might have that we can't get to.
And that's true anytime.
Okay, great. That's fabulous. So I want to thank everybody for participating in this webinar series, and a special thanks to our presenter for sharing her research with us. Also want to thank Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom and coordinating the technical aspects of the webinar. We could not do it without her. So next week, please join us for a webinar on water quality and wetland mapping. And following the Q&A session, we'll put a survey in the chat for you to give your feedback, and we appreciate any additional comments or feedback you have for the series. So now we're going to begin the Q&A session, and we will draw initial questions and comments from those that submitted in the Q&A box. We will do our best to get to all of them, but like I said, there are a lot. So I'm going to start with the ones that were upvoted. So the ones that more than one person had the same question. And so hopefully we can get started with that. All right. Well, let me find where I was going to start here. Okay, so Kevin initiated this question. We had a lot of people with the same one. What is the lifetime of these toxins in the environment after they are released from the cyanobacteria?
Well, that's something I'm really concerned about, that I was going to get a lot of questions that are kind of out of my area of expertise. I deal mainly with the animal effects. So there's probably other people in this call that know the answer to that better than I do. I know some things like cyanotoxin are very susceptible to break down quickly. And then I've heard that microcystin is very stable and can stay in the environment for quite a long time. But I do not know about any more information than that, so I apologize.
Okay, thank you. Another question: for many years, cyanobacteria and or certain cyanotoxins have been in the CCL list. Do you have any insights if these will ever be regulated for drinking water with national drinking water standards?
Yeah, that is not anything that I'm involved with. I just try to supply the information, but from my perspective, I think they probably will, but we just, as a general statement, not just for EPA, but for the world, we don't have a lot of cyanotoxin toxicologists. There's not nearly enough. And so a lot of the early work that was done was like wonderful, wonderful work, but it may not have, little important pieces of information that keeps it from being used for regulatory purposes. And it seemed like after that initial, like in the 90s, and, a lot of work was being done, it kind of died off as something that was more people were doing. So I think if they can get this sufficient research to guide what they need to do, it's possible there could be some regulations, or at least more guidelines, something more substantial than what we have right now.
Okay, thank you. I have one here. Is there a suggested time frame for testing to identify when an individual has been exposed to cyanotoxins? And can most labs do an intraperitoneal test?
Well, the intraperitoneal is like for injecting the toxin. So that's purely a research way of exposing the animal. But, I know with a lot of animal toxicities, if they can get the stomach contents, they will usually find it there. Unfortunately, the ones, I think, for wildlife and for domestic animals that are die quickly after exposure or found dead, they look at the stomach contents to try to get an idea of, and find the causative agent. things like antitoxin, it can be found in the urine. I'm not sure how long it can be found in the urine. It's cleared from the body very quickly. It's a small molecule, and I think it shoots through pretty fast, probably within 24 hours. And how you handle the sample, antitoxins they tend to break down, and you won't have evidence of them. But if they're in an acidic environment and out of light, they can last longer. So, I think that's why they're found in the stomach so well. Microcysteines are bigger. They probably can be, found longer, but I don't think we have, specific time points for those. But as soon as the animal's feeling sick, or the human it's always worth checking to see if you can find evidence. And the CDC has some urine tests. They're still working on developing some more, but they have some urine tests for identifying the cyanotoxins. But these still have to be done by labs with pretty decent chemistry capabilities. There's no little point of use test, as far as I know.
So related to that, as far as the breakdown goes, question is, will composting harvested cyanobacteria break down the toxins?
And now I think if you're destroying the cell, you're releasing the toxin, then I guess it just depends on how stable the toxin is in that environment. I don't know if that's actually ever been done. That might be a good study for someone. I know that in the sludge that, from water treatment facilities that's taken and put on, fields, you still have toxin content in some of those.
Okay. Got another question. Since it is difficult to accurately acute illness in humans, is there any work on increasing the voluntary reporting in HABs?
I was hoping by presenting it today that some people would be motivated, because 13 out of 50 states is not a good showing. Now, I don't know, 2020 is the most recent, so I don't know what 2021 and 2022 were like. Maybe it had improved, but I would hope that, I know it takes probably a lot of man hours. These are not just like simple little forms. There's a lot of information, but the more states that can participate in this voluntary reporting would be really helpful.
Yeah,
It helps all of us, with the feedback and kind of know what the patterns are.
Sure. And related to that, part of that question is also, is there a decent rate of ER doctors and veterinarians communicating to help accurate diagnosis of exposures?
That depends on the area. I'd say states that probably have more reported cases of it, the medical doctors and veterinarians are more aware. I don't think in North Carolina it would be on the top of anybody's list, because we just haven't had a lot of reports. We've had some antitoxin dog deaths, but they're, few and far between. And, I'm a veterinarian, and, when I don't think it's being emphasized as much as it should be.
Okay, thank you. Has there been any studies showing if there is an impact to air quality from these toxins being aerosoled?
There is a lot of concern about that. And they're in this current research round at EPA, I know that some aerosols are being tested in Cincinnati, and someone else proposed, I don't know, a research projects kind of apart from the general research planning, to look at inhalation of particles containing cyanotoxins. So I think the Office of Water and a lot of people are concerned about that, but there's not a lot of data right now, but people are starting the work on it.
For individuals who sample HABs, what sort of precautions do you recommend they take?
I definitely recommend, some general PPE. Like, you don't want it to touch your skin. I know last year or the year before, people were complaining, even people in the lab were complaining with some of the samples that came, from some blooms, causing their eyes to burn, and, it hurt to breathe. So I think each one's probably a little different, but I would start with gloves and definitely consider some respiratory protection.
Thank you. Got a question. What is being done about prevention and remediation and providing better guidance to state and local agencies? Michigan's residents are suffering under wasted costs of data monitoring and controlling cyanotoxins with outdated algicides like copper sulfate that makes the problem worse.
Yeah, unfortunately, that is not my area. So I can't comment on that.
Okay.
I know other people are adding things to the chat as well, because I see them pop up. So if there's a question that I've answered and I haven't answered it fully, there's other people have added some information in the chat.
Great, thank you. Has the EPA or any other sources to your knowledge done any research into bioaccumulative effects of cyanotoxins?
Oh, probably bioaccumulation. And yeah, I mean, the marine toxins, that's how they cause a problem. They're bioaccumulated, and like the shellfish. There's been some work for freshwater as well, but it definitely lags behind what we have for marine. I would think microsystems are tough. What I've heard from chemists, they're tough to measure once they're in like a protein medium, so you don't know if you're always getting the accurate amount. But yeah, I know Jim Lazorchak's on the call. I saw, and he probably might have some information. He has done some, he's looked at the literature on, accumulation in fish muscle, because that's another big concern for OW, and we need more work on that as well.
Okay. The EPA has a handful of methods for the quantification of select cyanotoxins and environmental matrices like freshwater. Considering this talk is focused on HABs effects on mammals, what is the current outlook on clinical matrices like blood, urine, serum across various cyanotoxins? Well, I think that's more of the CDC's approach. That's their focus on this. And I know the chemists or a large group of people in the EPA are currently working on sampling and testing of benthic samples, since it can be different from the planktonic. But this is just sampling the waters that they're focused on. So as far as urine and blood, I think the CDC would probably take that on. And I do contribute samples from my test animals for their validation of their test when they've already requested some of the anatoxin urine as well. So we had somebody comment that it's intriguing that you saw such marked differences between males and females. Can you explain or speculate why? No, I do not know why. And it could be, like the way they're metabolized differently. And can be differences with the cytochrome P450 in the livers of males and females. That can be different. But no, we finished that and got it published. And then there was COVID, and the toxicologist retired, and we moved on to anatoxin. So that is something I would like to explore, and maybe have to do it through transcriptomics. Maybe we can look for it that way. I have a question about, do the toxins have some metabolic function in the cyanobacteria, or is it a defense mechanism?
That again is not my area of expertise, but you would think if an organism is going to produce something, there's a purpose, so I mean, that's the way people look at it. It's likely a defense mechanism in a lot of cases, but that's not my area.
What about why the Great Lakes seem so vulnerable to HABs? Any comments on that?
Well, I know the area that, in Lake Erie that gets affected so much is relatively shallow, that southwest end of it. But beyond that, I cannot speak to it.
Do you have any thoughts on how toxicologists might identify meaningful thresholds for toxins / HABs that are primarily benthic mats?
Well, it's identifying the toxins that are in that, and then doing the same work, for that toxin. And like I said, liver toxins, that's more straightforward. You've got a lot of biomarkers that you can measure and look at. You can look at histopathology. Neurotoxins, like I said, we're still struggling to find good endpoints that we can measure with the neurotoxins. So maybe after I finish an antitoxin study, we'll know more, but and dihydroantitoxin will definitely be in everybody's future. So, I feel like that's something we'll be doing work on in the future. But right now, that's, dihydroantitoxin it changes its form, and there's a lot of variation in how it can be presented, and so it's kind of tough to know how to do research on that. Antitoxin A is a little more straightforward.
Do we know what triggers production of microcystin?
No, again, not my area. I'm sorry. I only deal with the health effects in animals.
Do you do any microbiome analysis in oral experiments?
I have not, but I think it's very valid to consider that.
What are some of the threshold water conditions that these HABs are expected to strive best in terms of chemical water parameters like temperature, DO, conductivity, pH, etc.?
I'm sorry, I can't answer that.
Okay. Are there examples of guide values for some wild animals?
No, the value, as far as EPA is concerned, it's the guidance is all we have is what I showed in this presentation. It's for humans, and it's for microcystin LR and cylindrous spermatocin. And it may be, Lee Jones or somebody would know for the wildlife things, but I think they don't have any more to go on.
Is there any evidence of clams being used to filter the water?
That I don't know about. I know they look at, they use different filter feeders to measure deprivation studies, and how long it takes before they're safe to eat. But I don't know about that. I see some people are commenting on that in the chat.
Okay, let's see here. Were the mice used in the oral exposure study, the PPAR alpha knockouts?
No, that's something that's used with PFAS work, but I have not. I don't think that's a factor we've considered with the cyanotoxins.
Okay, question, is the food / dietary supplement referring to spirulina.
Spirulina,
Thank you.
spirulina, or spirulina. Yeah, I mean, that's a big one. I know that there's lots of other supplements, and I don't know if it was the spirulina, but a lot of pet and horse supplements, contain algae that may be contaminated with cyanotoxins.
Okay, we had another question about just safety. I think that's already been answered, so I won't go on to that. Is cyanotoxin poisoning of animals mandated reporting in any states? And if so, do you see higher numbers reported from those states? And can we use that to extrapolate?
I don't think it's mandated anywhere. I have talked to several people in different states, and some people don't even, they may or may not know that they can reach out, or people talk to other people, and that's how information gets back to me or to someone else, and then I hear of it. But I don't think there's mandatory reporting. Even if there was mandatory reporting, it would probably be the CDC, and they don't, it's a voluntary reporting system they have with the OHABs.
I know there are no drinking water regulations for cyanotoxins. Is there a good way for people to check if their public drinking water has these toxins?
Well, sorry, I'm not sure how to answer that, because that's not an area I deal with. I think different municipalities, well, the EPA, because of the unregulated contaminant monitoring rule, the larger water systems they do periodically test and report and participate in that. And then I think that is accessible on the EPA website. But as far as like individual towns and all, you just have to contact, I guess, your water plants and see what they have. I honestly don't know.
Can you comment on the carcinogenicity of cyanotoxins?
That is always something that's evaluated. So EPA does a health effects statement document for each cyanotoxin when they feel like they have enough information, and that is always a consideration. And it's probably the most limited amount of data on cyanotoxins, because up to this point, it takes, that's a very long-term study, and people are looking for ways to shorten that so you can screen more chemicals for their potential to cause cancer.
Could you share any resources or information you have regarding dog or livestock toxin exposure thresholds? And I know a couple of people kind of added some of their resources in there, but if you have something specifically to add, that would be great.
I do not. I would look and see what the people from the different states are putting in.
Okay.
I'm sorry, I don't have anything more or that, than what I've already presented.
Do toxins bioaccumulate like in fish?
Yeah, that's something that is really people are curious about, having more information on that. And it's similar to what I answered earlier. There's some work looking at the bioaccumulation, and it is ongoing. It does, for most of the papers I've looked at, it seems like it accumulates in the liver and less in the muscle tissue in most cases. So that's good. But yeah, that's not an area that is well established right now.
Okay, well, I think by my clock, we have time for one more question. What sampling method did you use to test different waters in the state?
Okay, I don't test the waters. I buy toxins from sources and, you know, the standards and use them. I never leave the lab. So not a field person.
Okay. Well, between the Q&A and the chat, there's a lot of discussion going on, a lot of resources being shared, and a lot of
Good.
gratitude for your presentation today. So we really appreciate your time. And I know I did not get to everybody, and I apologize, but we kind of saw when all the questions started coming in that was going to be a problem. So please use Donna's email that is being shown on the screen right now, and forward any questions or follow-ups to any questions that were discussed today. And we really thank you for your time, Donna, your expertise, and we look forward to seeing all of you at the next webinar.
Well, thanks. And people, please reach out. I love to talk about this, and I learn a lot when I talk to other people. So don't hesitate.
Very good. Thank you.
All right. Thanks a lot. I enjoyed it.